This screencast is covering two examples of moving molecules across membranes. So we're going to look at a detailed example of facilitated diffusion. We're going to look at the potassium channel. And we're also going to look at a detailed example of active transport, the sodium-potassium pump. So this is one of those protein pumps that are moving things against their concentration gradient. So these are two specific examples. One is passive transport, facilitated diffusion. That's my potassium channel. And then the other one is active transport, and that's the sodium-potassium pump. The reason why we're going to look at these is because you're going to need to know them for when we start doing neurons and neurobiology, because these are very important channels and pumps for how we can send information down a neuron. But both of these channels and pumps are used in a lot of other places, as you saw one in the small intestine in a previous screencast. So it's not limited to here, but we will learn about them in more detail even as we get to doing neurons and synapses. Okay, so here is a potassium channel. And this particular potassium channel is a gated channel. It's going to open only at certain times. And you can see the membrane in a neuron, in this axon, we've got it's positive on the outside when it's, outside the cell, and negative inside the cell. Okay, so this is the membrane. This is inside the cell. This is outside the cell. This is the cell membrane. Okay, and the structure of the potassium channel has this ridges on right out here on both sides that are positively charged. The amino acids that make up those that part of that protein on the channel is positively charged. And because when a neuron is at rest, it's very negative on the inside and very positive on the outside. The positive charge part of this protein is attracted to the negative inside of the neuron. So you can see this is where it is happy and at rest, because opposites attract. Well, when a neuron is firing, it actually flips its charge. It becomes negative on the outside and positive on the inside. And because of that flip, then all of a sudden this positive part no longer wants to be here, it wants to be on the outside. So you can see it's going to shift from down below where it used to be positive to negative, and now it's going to go up here where it's actually negatively charged now. And that causes the potassium channel to actually open, okay? This is the part that you need to be able to explain. There's some other funky ball at the end that can kind of stop the ions from flowing through, but these first two, the channel closed is because the positive amino acids on that protein channel are attracted to the negative inside of the neuron. When that neuron fires, when the signal's being sent down the axon of a neuron, then those charges on the outside and inside of the cell are going to change place. So these positive amino acids are going to be now attracted to the outside, which then opens up that potassium channel. So this is a facilitated diffusion because these are ions that are moving through the membrane. It's going along its concentration gradient through passive transport, and but it has to go through a protein, so it makes it facilitated diffusion. The active transport that you need to know is the sodium-potassium pump. And I showed you a brief animation on that before, and I'm actually going to pull that up again just to kind of give you the steps of it. So again, in a neuron, and this also was for the small intestine as well. You saw that was the example I had given you before. And that the outside is where we're going to pump sodium ions. Okay. The inside is where we want to take the sodium ions, and they're in lower concentration here, but we want to get a really high concentration of sodiums outside in the extracellular fluid. Outside the cell, it's also very low in potassium ions. So we're going to actually keep moving potassium ions in and moving sodium ions out. So we're moving them both against their concentration gradient. So sodiums are high here and low here, but we're going to move them from low to high. And the potassiums, they're low here and high here, and we're going to move them from low to high. Okay? So sodiums are going to go in, potassiums are going to go out. When the protein pump, the sodium potassium pump, when it's at rest, it's open to the inside of the cell. And you can see the shape that it has here. It's perfect to fit 3 sodium ions. And the sodium ions, through kinetic molecular, will end up, binding to this protein. The next step is that an ATP molecule will come and phosphorylate, so drops off that phosphate like I showed you on a previous slide. It's going to drop off a phosphate and now become ADP. This is giving that protein energy. That's enough energy for it to change shape. So now it has changed shape and it's open up to the outside. And the sodium, and it actually expels those sodium ions. You can see it changes shape on those sites where they bound anyway. But it's going to then open up the potassium binding sites. So potassium through kinetic molecular will end up binding to the inside. And then what happens is once those bind, that phosphate gets released and the protein changes shape again back to its resting state. So the potassium can then be expelled to the other side of the cell. I'm going to show you a quick animation of that happening, and I'm going to try to talk over that animation so you can kind of see what's going on. Okay, so here is the sodium potassium pump, and you see 3 sodiums go in. And did you notice right there that that was the ATP phosphorylating the protein? Okay, and that allows it to change shape to be open to the outside. And then the sodium gets released, the potassium comes in, and then gets released to the inside. So you can see as it goes, when it's open, it's at rest. That's now the sodium is going to go in, and you see this ATP come in here. It's going to phosphorylate, drops off the phosphate, and that gives it the energy to change shape. The sodium will leave, the potassium will come in, and when the potassium now comes in, this phosphate gets released and it reopens up to the inside. Okay? These steps are pretty straightforward. Hopefully you'll take a look and make sure you can work your way through it. All right, so that's our sodium potassium pump.